Hi, this is our Biochem lab poster and it's by Krystina, RAHIL, Moses, and CLAUDIA. So, we have hypothesized that if you treat maize with EMS, it may cause actin patches and mutants to be larger in ratio to the guard mother cell than in wild types. As you can see in picture 1, the actin patches line almost perfectly with the guard mother cell and are at equal lengths on either side. Now, if you look at the pictures of the mutants on the right, you can see actin patches longer than the guard mother cell, as well as thicker and sometimes asymmetric. We are looking specifically at how long these were in comparison to the wild types. So to start the process, we had to phenotype and dissect the leaves. We used various materials listed here to dissect the plant and prepare it for phenotyping. We labeled the plants and a corresponding microscope slide, then a leaf was removed from the plant and attached via superglue to the side. Approximately 5 leaves were glued to each side. Following, we removed the leaf from the slide after the glue had dried. Once the leaf was removed, we viewed them under the microscope and recorded the phenotypes. After we performed leaf dissections as directed by the TA to prepare for the next phase of the project. These pictures show slides from the Zoe fluorescent microscope of 2 maize plants from the family U2293. The pictures show plants 6 on the left and 8 on the right, which are both wild types. You can tell that they're both wild types due to there being no defects in the subsidiary and guard mother cells of the plants. These pictures are from the same family U2293, number 10 on the left and number 25 on the right. However, as you can see compared to the previous slide, many of the guard mother cells and the subsidiary cells are abnormal and misshapen and do not look like the wild types in the previous slide. We have phenotyped these plants as mutant maize plants.
The overall purpose of this lab was to stain the actin and the DNA. After phenotyping, the asymmetric and symmetric leaf zones were dissected out and placed into a 15 milliliter Falcon tube. 1 milliliter of prefix solution was added for 30 minutes to open the tissue and prepare it for fixation. The prefix solution is then taken out and 1 milliliter of fixative solution was added for 60 minutes to stop the opening and preserve the tissues. After an hour, the fixative solution was taken out and 1 milliliter of wash solution was added for 10 seconds and taken out, which was followed by 1 milliliter of extraction solution for 15 minutes, which gives more holes in the membrane. After 15 minutes has passed, the extraction solution was taken out and the Falcon tube was wrapped in foil to prepare it for the next solutions, which are light sensitive. After being wrapped in foil, 1 milliliter of phalloidin solution was added for an hour. After an hour, the phalloidin solution was taken out and 1 milliliter wash was added for 10 seconds and then taken out. 1 milliliter of propidium iodide solution was then added for 10 minutes while the Falcon tube was still wrapped in foil. After 10 minutes, propidium iodide was taken out and 1 milliliter of wash solution was added and kept in the tube. All the materials that were used are listed to the left. The prefix solution, as stated before, is to get the tissue open and ready for fixation. The PME is a buffer that contains pipes, magnesium sulfide, EGTA, water, and with a pH of 6.8 with sodium hydroxide. Triton is a detergent and surfactant that helps with surface tension and adhesion problems. MBS is an actin crosslinker that helps keep actin together, and DMSO pokes holes in the lipid bilayer. The fixative solution's purpose is to open and preserve the tissues. The wash solution's purpose is to take out and stop whatever solution was before it. The extract solution's purpose puts more holes into the lipid bilayer. The phalloidin solution is sensitive to light and was discovered in the death cap mushroom. Its purpose is to stain for. It attaches to AF488 fluorescent protein, and when hit with excitation light, it gives off a wavelength. And finally, the propidium iodide solution is also sensitive to light, but stains for DNA when hit with light. It gives off another wavelength.
Okay. Now, analyzing the confocal images of our plants, you see from plant 6 is obviously a wild type phenotype. One thing about this plant is that it's very advanced. which may explain like the larger numbers we got when compiling this data. The actin patches are very large. You can see them pretty clearly, and you can even see like the stoma as it's already splitting up. So on plant 10, you look close. Obviously, a mutant. It's not very uniform. It just looks abnormal overall. The nucleuses are just wherever they want to be. The actin patches are, in contrast to the wild type, actin patches are smaller, and for plant 8, it's just like plant number 6, except it's not as advanced, but it's still a normal wild type. So nothing wrong with the subsidiary cells, and everything's just in line. For plant number 25, this is also a mutant. And you can see a lot of the actin patches, but if you look closely at like the nuclei and everything, how it's split up, it's also very abnormal. And then considering how we actually measured the cells when compiling our data, we use something called Fiji or ImageJ. It's available for anybody to use. It's a program that let us analyze these confocal images. And what we did to measure, we basically measured the length of the left actin patch, the actual guard mother cell, and as well as the right actin patch. So the reason why we measured the middle of the guard mother cell is that so we can use this data and compare it to any other cell in any stage on any plant, basically.
Actin patch length measurements range from 0.9 micrometer to 8.3 micrometer, with the average length of the left actin patch measurement being 3.0 micrometer, and the average length of the middle of the guard mother cell measurement being 3.7 micrometer, and the average length of the right actin patch measurement being 3.6 micrometer. There is a variation between the average left to middle ratio and the right to middle ratio, with a difference of 0.08 micrometer. From the data presented, we observed that plant 6, bottom half, is at very advanced stage of maturation. Therefore, it has larger length for the actin patches and the guard mother cell. Overall, the actin patch length measurements range from 5.3 micrometers to 18 micrometers, with an average left length measurement of 11.5 micrometers, an average middle length measurement of 12.1 micrometer, and an average right measurement of 0.96 micrometer. The wild type had a smaller difference in average, which is left to middle ratio, and average right to middle ratio. This difference only came to about 0.02 micrometer. The paragraph displays the average length of the actin patches in mutant and wild types. From the data we have collected, presented in blue is the mutant plants, and red is the wild type plants. We can see, measured in micrometers, that the length of the wild type actin patches are greater in size compared to the mutant. The wild types have a larger left than right actin patches when compared to the middle length, where the mutant has a larger right than left actin patches when compared to the middle length.
The mutant plants number 25 and number 10 did not support our hypothesis completely. When treated with EMS, the left and right actin patch lengths were affected. The lengths were smaller on average when compared to their guard mother cell length. The wild type plants number 10 and number 8 supported our hypothesis. The data we collected shows that the ratio of the length of the left actin patch over the length of the middle of the guard mother cell versus the length of the right actin patch over the length of the middle of the guard mother cell being very similar and not varying in ratio.